Hi, I'm Evgenia, a postdoc in the Professor Sir David Kleinerman lab at the University of Cambridge. Today, I would like to present the main research highlight of our brain paper entitled Imaging Protein Aggregates in the Serum and Cerebrospinal Fluid in Parkinson's Disease. The focus of our work is development of early-stage diagnostic biomarkers in PD based on blood and CSF using state-of-the-art single-molecule imaging tools available in our lab. In our paper, we perform single-molecule imaging of aggregates before and after immunodepletion of either alpha-synuclein or A-beta from respective biofluid with antibodies using conventional magnetic bead-based immunoprecipitation assay. Once the supernatant of each immunodepleted fraction is collected, we image it with Aptima DNA paint, or briefly AD paint. This super-resolution fluorescent technique is based on detection of aggregates electrostatically absorbed onto twin-coated glass surface with the aptamer. This aptamer is conjugated with a docking strand and has a complementary imaging strand freely diffusing in solution and generate a blinking upon transient binding to the docking strand, as shown on this video. By recording individual blinkers over 1,000 frames, we could super-resolve our aggregate and precisely measure their size and shape. Since our aptamer bind to both alpha-synuclein and A-beta oligomers and fibrils, we cannot distinguish between these two. Therefore, in our experiment, we measure 3 wells per each patient, as shown here on an example of PD serum. Even by eye, you can see a visible reduction in the number of blinkers in both immunodepleted fraction, which shows a proof of principle of our approach. And the difference between need and each immunodepleted fraction will give us a measure of relative alpha-synuclein and A-beta aggregate levels, from where we could easily calculate alpha-synuclein to A-beta ratio. In our paper, we established whether this ratio could serve as a useful diagnostic biomarker in PD. For this, we apply our approach to a homogeneous group of serum samples from PD patient within 2 years of diagnosis, together with age and gender-matched healthy controls, and found that the ratio of alpha-synuclein to A-beta aggregates is significantly increased in PD compared to healthy control serum, and that this ratio, as well as relative alpha-synuclein levels alone, discriminate ED from healthy control cases with the accuracy of almost 100%, making it as a promising diagnostic biomarker. Since we perform super-resolution imaging, we are able to measure the size of aggregate and found that serum aggregates are surprisingly large, ranging in size from 20 to 200 nanometers, with a higher proportion of longer alpha-synuclein aggregates in PD, but not A-beta, which shows similar sizes between PD and healthy control cases. As for CSF, we found that the concentration of aggregate in this biofluid is almost 20 times smaller than in serum, which was under detection for our immunodepletion experiment. Therefore, only AD paint imaging was performed, from where we found that there was no difference in the total number of aggregate between PD and healthy control CSF, but again, there was a higher proportion of longer aggregates in PD. Also, aggregate in CSF were in general smaller than in serum. Thank you very much for your attention, and I would like to acknowledge all our funding bodies supporting this research. Bye.